Hello, my name is Stanley Liphadzi from the Water Research Commission. I'm responsible for heading the research and development branch or portfolio in the Water Research Commission. The Water Research Commission in May this year announced and also launched a very important program which is about using the wastewater or sanitation to monitor the COVID-19 spread in South Africa. And we introduced this program, or we launched this program, because we realized that having only clinical trials will actually take a long time. And the wastewater-based sanitation program actually will assist us, or this epidemiological program will assist us to quicken the tracking of the virus or the infection of the virus in our communities, because it focuses on the whole community, it focuses on the whole city or on the whole area. And then for us actually, if we can find the presence of the genomic or the signature of this virus, it will actually assist us to realize or to realize that there is actually a challenge of the COVID-19 in that particular community. And the data also can also assist us even to estimate the number of people that are infected by the virus in a specific area or a specific community. So the WRC, when it launched this program, we decided that we would want to have it in a stage kind of phases. So the first stage or the first phase, it is about the proof of concept. And by proof of concept, we would want to test the protocols relating to sampling, relating to testing of the virus or of the samples for the virus in the laboratories, and also to see if the data or the test result that you are getting are making sense for us to be able to use for decision making. So that's what the proof of concept is actually meant to assist us to do. So from there, once we establish that and we are happy with the outcome or the results from the proof of concept, we'll move to the second phase, which will be the pilot monitoring of the virus in the samples or in the wastewater systems. So we'll choose specific areas. At the present moment, our priority will be looking at the epicenters, which are the hotspots of the COVID-19, different provinces like Gauteng, Western Cape, Eastern Cape, and KwaZulu-Natal. So we know about the hotspots in that particular areas. And then we will sample specific points of the waste treatment, of the waste system or the wastewater systems. We will also look at some areas that have got non-sewer systems as well, and then we'll choose some strategic points, and then we'll sample from that, and then we will test to see if we can find the signatures or the RNA in those particular samples. And then we will also use that data even to test drive some of the questions, in answering some of the questions that we really have. For example, does temperature impact the state of the virus signature in the sample? What about the location? What about the land use? For example, is there any difference in getting data from school as versus getting the data from the community itself? Can one actually correlate and see the relationship from that? But again, at the same time, we can also use different places or land uses also to see where the spread actually of the disease may even go faster. You go to jail as well. Samples from the wastewater coming from the jail, you find, you compare it with samples that come from schools, and you find that you also compare that with that from the shopping centers. Surely there may be a difference depending on what people do there and what kind of measures that they have put in place to prevent the spread of the disease. And we'll check all that. And then from there, once we've established and we've answered all those questions, we'll even try to plot the data in different platforms as well, like the GIS and even other platforms that may be useful for decision makers. And once we are certain and we are satisfied with that, then we can move to the national surveillance stage or monitoring of our program. Then that's what is like the ultimate prize in terms of the program. That's what we would want actually to have for the country and even for the continent. And once we have we get into the surveillance monitoring program, we'll need many stakeholders now. We'll sample many places now. We will try to get as many people as possible and many students as possible to assist us in sampling, to assist us also in funding the testing of these samples. And then we will gather all this data and establish the platform for more intelligence generation out of that. So we'll actually have to have more information, more data, so that we can be able to make decisions that are much more clear for the Department of Health, for the Department of Water and Sanitation, and even for other people who may want to use that data or that information. And the other important thing of us having this surveillance program will be that we can also introduce other pollutions or pollutants that we want to monitor in our systems, like the heavy metals, even other diseases like polio and so on. We can also introduce monitoring of other things